In this video, we are going to talk about insulin and the way of production of human insulin. If you read the text from here, you will see that diabetes are the people who has some problem with the production of the insulin. This problem may have been originated from the destruction of the cells in pancreas. So pancreas cells have some problem for secretion of hormone, and that's why a diabetes person doesn't produce enough insulin. So the treatment is by injecting some insulin into the blood. So that's why you will see the people having diabetes they need always to do some injection to their blood. In the past, insulin was extracted from animals. For example, porcine and bovine. There are two types of insulin extracted from pancreases of pigs. Porcine from pigs and bovine from cattle. And they have been widely used. In terms of the different amino acids in the amino acid sequence, porcine, which is coming from the pig, has only one difference in the sequence of amino acid comparing to human. And bovine insulin has three differences. So there are still amino acid sequence differences for these two types of insulin produced from the animals. There is another insulin extracted from shark insulin. This has been used for diabetics in Japan, and it has 17 differences. So more differences comparing to porcine and bovine. However, despite these differences, these insulin could bind to the receptors and cause the lowering blood glucose concentration. That's the function of the insulin. But some of these diabetics, some of these people that they used animal insulin showed or developed some allergy to these animal insulins. And because of that, it happened that because of these allergies, it happened that the science decided to find out another way, which were successful in 1982, that human insulin was developed and was commercially available for the first time of the human life. How did it happen? It is just because of the genetically modified E. coli bacteria. So we are going to explain how the genetics and how this technique could help the massive production of the human insulin, which is completely the same as human insulin that was produced by human and is vastly are used nowadays. What is the genetic code as universal? Pause the video and think about it. What was your answer? Yes, I can guess that you answered correctly. Genetic code is universal means that all the bases are the same and the genetic code are the same in all living things. That's the meaning of being universal. and each codon produces the same amino acids in transcription and translation regardless of the species. Okay, regardless of the species, no matter what the species is that, the codon is the same and the amino acid is the same. So the same process, the same procedure, and the same codons. So you can see that the sequence of amino acids in the polypeptide remain unchanged. Therefore, we can take genes from one species and insert them into the genome of another species. And this is the process of gene modification or gene transfer using a bacterial E. coli. This diagram shows how a bacterium can be used for genetic transfer. So as you can see, there is a bacterial picture over here. Actually, the plasmid should be a little smaller. So if I want to draw a bacteria showing DNA in a better proportion comparing to the size of the plasmid, that would be something like this. So a bacterium has the naked DNA without any cytoplasm, without any membrane, and another DNA which is called plasmid. So this plasmid has a key role in genetic engineering. The technique includes the exposure of the plasmids from the bacteria into some enzyme that cut the plasmid. So this is the place that the enzyme will act to cut the plasmid, and then the gene, which is in here, insulin gene, will be inserted into the plasmid as the step 3, and then this engineered plasmid will transfer again to the culture, to the bacterium, another bacterium, the same E. coli, not this E. coli. I mean, the culture of the E. coli will receive this bacteria, including the new plasmid, and then this bacterium starts to grow and starts to increase the number in a medium culture by massive production of the bacteria, and all of them includes the new plasmid having the gene of the insulin. And then what happens is that the bacteria start to produce the protein because it has the gene over here. So they are producing the protein of the insulin. Here is another diagram showing how the restriction enzyme will cut the DNA and extract or remove the desired gene, which is here, and then with the presence of the desired gene and the plasmid, which has been cut with the same restriction enzyme, so the presence of this desired gene and the plasmid that has already been cut with the presence of another enzyme which is called ligase, which is responsible for attaching this gene into this area. Then finally we will have the plasmid including the new gene, and then this plasmid including the new gene will can be inserted into E. coli, And then this E. coli can be cultured in a medium in order to mass production. And once we have an E. coli with a new gene, with the plasmid having new gene, it is possible to produce a large amount of the insulin. That's it. I hope you enjoyed the lesson. And this is the end of 2.7.